So I'm from the Institute of Mathematical Sciences, as Sunil pointed out. And I must thank the organizers first, Ranjith, Dimple, and Mithun, so for inviting me. And let me be very clear at the outset, I'm not a chromatin expert, so it was a very good learning experience for me listening to the stalwarts and the experts in the field. And I hope that I will take that learning and implement some of it in my own research. So let me begin with this slide with this cartoon which many people have shown in different incarnations, which says how the genome is basically organized in an hierarchical fashion to solve the packaging problem. And many of the experts have talked about it, so I will not go into too much detail on it. But we have heard a lot of talks focusing on different experiments on how to probe the different scales of organization that you see. But in parallel, actually, during the last decade or so, there has been a lot of different computational models that have been developed, and these come in different resolutions. And this has sort of enabled us to get a better understanding of the genome organization at different scales, ranging from what happens at the level of the nucleosome to subnucleosome, supranucleosome, and so on. And these are some of the examples. So this is from this very nice review article where they have sort of summarized some of the models that people have been using. So the one I will be focusing on is basically of this resolution, which is near atomistic to atomistic, which helps you to look at the dynamics of the nucleosomes. So what got us really interested into this problem is this unwrapping transition of nucleosomes. So nucleosomes, as you know, in here, you could see some section of the DNA unwrapping from the histone core, and this is what sort of gives it access to the various kinds of transcription machineries. And let me summarize some of the very seminal experiments that people have done in this field. So the one that comes to mind is the seminal work from Carlos Bustamante's group, where they were able to look at the force extension profiles of how a mononucleosome unwraps. So what they pointed out is there are basically two regimes, where the low force regime at about 3 to 5 piconewtons, where the outer turn of the nucleosome unwraps, and as you go to higher and higher forces, the inner turn starts to unwrap. And following that, they have a very recent work in PNAS, where they have focused also on some subnucleosomal particles. And there was another very interesting work from Takjip Ha's group, where they pointed out, in fact, this unwrapping that you see under force is in fact asymmetric. For example, if I have a DNA sequence like this, where one of the ends is more flexible as opposed to the other, it's actually the stiffer end that comes out first, because it is not really able to accommodate that turn. And subsequently, there has been very nice works from the groups of Louis Pollack and co-workers, where they have sort of used time resolved Sachs experiments at different salt concentrations. And what they pointed out is, in fact, the salt-induced disassembly of nucleosome can involve multiple pathways. You can have asymmetrically unwrapped states, or even the ones which look symmetrically unwrapped. In fact, why do we were not really the first ones to really think about the nucleosome. There has been a lot of very exciting work in this area. So let me point out some atomistic simulations that people have done on this, and it really is a very challenging feat. So the first one that comes to mind is this group, it's the work from Axiomentiv's group, where in order to look at the nucleosome unwrapping, what they did was they looked at different, they did simulations at different salt concentrations. As you see at 0.5 molar, they don't see any unwrapping event. But as the salt concentration is increased, like to something which is, this is quite unphysical, like 1 molar magnesium chloride and 3 molar sodium chloride, but they do see some partial unwrapping happening. And these simulations were done on this highly specialized Anton 2 supercomputer, which is maintained at DE Shaw Research. And very recently, there were much longer simulations that were carried out by Shaitan and co-workers, where they were able to see even more unwrapping events. But of course, all of us do not have access to Anton 2. We are poor, so we had to come out with a different strategy of looking at this problem. So what we did is we took a coarse-graining route. So we developed this force field called coarse-grained force field for energy estimation. And as you may have realized, it's basically an acronym for coffee. So we called it coffee. And you might ask, like, why was the force field called coffee in the first place? So the simple answer would be, we were simply following the tradition, of naming force fields after different kinds of drinks. So you have the martini force field, you have the absinthe force field from Rohit Papu and co-workers, and you also have what is called lassi. But surprisingly, no one had named the force fields coffee, which is in fact, a popular drink among theoreticians. So in a good cup of coffee, what you have is essentially a judicious combination of different ingredients, right? You have a probably nice coffee beans, sugar, and milk. And this is exactly the philosophy we had for developing coffee. So here, basically, what we combined is a coarse-grained model for DNA and a coarse-grained model of protein. So we did not want to do too many tweaks. In fact, we made no tweaks to the individual force fields, and we wanted to combine them in a proper way so that we get a transferable force field for looking at different DNA protein complexes. Now, let me just, I will talk about this slide very briefly. So let's say this is an atomistic representation of a given, let's say, a protein in water, where you see all the atoms are represented explicitly. So in coarse-grained models, what you have is you have a much simplified representation. You retain what are probably the relevant degrees of freedom. And in this exercise, what happens is your underlying energy landscape becomes much more smoother. You get rid of the irrelevant minima here, as you see. And once you have a less bumpy landscape, the sampling becomes much easier. So you can go to much longer time scales and much more relevant length scales. So let's talk about this first ingredient in our model, which is this 3 interaction site model of DNA, which we have discussed in this paper. So here what we have is each of the nucleotides in the DNA is represented using 3 interaction sites. So one which is placed on the sugar, one which is placed on the base, and one which is placed on the phosphate. And this is the energy function, as you see. It has the typical terms for bonded interactions, angular interactions, excluded volume, electrostatics, and perhaps the key ones here are the stacking and hydrogen bonding terms, and these are all sequence dependent. So I will not go into how we sort of calibrated these interactions, but if you're interested, you can see this reference. But what we showed, it basically does a very good job for both single-stranded DNA and double-stranded DNA. So we were able to reproduce the effective scaling exponents for single-stranded DNA for poly-DA and poly-DT. So as you see, the ones from experiments are shown in open symbols, and the ones we estimate are shown in these circles. And similarly, we were able to also reproduce the melting temperature for different hairpin sequences. So here the comparisons are shown here in this table. Now coming to the second ingredient, this is the self-organized polymer model for proteins, where here again, as you see, each amino acid is represented using 2 beads, one which is on the backbone and one which is on the center of mass of the side chain. And our group had extensively worked on it. So this is the energy function. As you see, it has the standard terms for bond connectivity, excluded volume, repulsion. And the key terms are these 3, which describe how the side chain, and the C alpha beads, and the C alpha side chain interactions term looks like. And of course, you also have this electrostatics, which describes how the different charged amino acids interact. And the important part is this part of the potential is the one which encodes the sequence specificity. And this has been extensively used by Govardhan Reddy and others to look at folding of different kinds of proteins, like the green fluorescent protein, ubiquitin, and so on. Now, how did we go about combining these 2 models? So we can write down the protein-DNA interactions as basically a combination of the TIS DNA potential for DNA, the SOP-AC for the protein, and then we also have a term for the DNA-protein interactions. And naively, one can write the DNA-protein interactions as coming from electrostatic component and a non-electrostatic component. So the electrostatic component here is treated using Debayan theory, and then the non-electrostatic component you can write as a combination of the DNA-protein contact potential, which is based on basically the native structure, and you also have a component which is completely repulsive, and these define the non-native interactions in your system. And so this is how the native interaction potential looks like. It's basically a Lennard-Jones potential. And the important part here is this epsilon xy. So that defines the energy scale for the interactions between the different, the amino acids and the nucleotides. And the way we went about calculating it is basically by collecting this contact statistics from a PDB database. So the energy scale is basically just related to the free energy cost of forming a contact between residues X and Y. And this is how we can define it. So this is the probability that a contact exists between X and Y of residues type I and J. And this is just corresponding to the random occurrence of X and Y. And these kind of potentials have been thoroughly discussed in the pioneering works of Shiraga, Tanaka, and others. Now, and this is just the table that we get from this exercise. As you see that the range is quite a bit for the different interaction energy scales. In fact, the strongest interaction is between guanine and arginine, as you can see from this scale. Okay. So the first thing we try to do is how we are doing with respect to getting the Sachs curve for this nucleosome sequence. So this is one with the alpha satellite DNA sequence, and we had a very high resolution crystal structure for this, which was reported by Richmond and co-workers. And in fact, this is, I think, the only one where you also can resolve the histone tails. So as you can see, the theoretical Sachs curve, which we computed, which is shown here with this red line, is pretty much on top of the experimental curve, and the radius of gyration that we estimate are also in quite good agreement. And next, we wanted to find out is what happens at different salt concentrations. Are we sort of able to recapitulate some of the observations that people have seen from experiments? So we've looked at the salt concentration range from 0.1 molar to about 1 molar, and as you see, like when you're at low salt concentration, the nucleosome is fully wrapped. However, as you keep increasing the salt concentrations, you populate various partially unwrapped states, and you see both of these populations. You see states where it's asymmetrically unwrapped, and you also find states where it's symmetrically unwrapped. And the next thing we looked at is what happens if we chop off the histone tails. Interestingly, what we find is once you chop off the tails, you always see some bit of unwrapping, even at low salt concentrations. And as you increase the salt concentration, you typically see more and more unwrapping. And interestingly, at very high salt concentration, we saw there was a complete disassembly of the histone core from the DNA. And this is just showing the average number of unwrapped base pairs as a function of salt concentrations. And you see that it's always higher for the situation when you don't have the tails. How much? 10 seconds. Okay. And we also wanted to find like some idea about the asymmetry. So the way we have defined the asymmetry is based on this ratio, like the number of base pairs that are unwrapped from the left side and the right side. So interestingly, what we find is if we are at this 0.2 molar salt concentration, there is not really much of a preference between the asymmetric versus the asymmetrically unwrapped states. But as you go to something like 0.5 molar, one side sort of starts to win. Interestingly, when you chop off the histone tails, you do not see that bit of asymmetry. So that signature of asymmetry is lost as you chop off the histone tails. Now, the other aspect we looked at is whether what happens if you mutate some of these residues at key superhelical locations. So we know that these arginine phosphate salt bridges are key to nucleosome stability. So what we wanted to try is what happens if we mutate some of these arginines to lysines. Interestingly, what we find is, as compared to the canonical nucleosome, so we looked at 0.5 molar, you do see more and more unwrapping. So there's a major peak that corresponds to about 90 unwrapped base pairs. And what I wanted to point out is this is, of course, as you will know, it's a largely chemical effect, and this is not accounted for by other existing coarse-grained models that exist. And so in a new version of our model, we have also sort of included ions explicitly, so I will not discuss this because of lack of time. And this is the summary. So we have a robust framework where we have combined the coarse-grained potentials for DNA and proteins. We are able to recapitulate some of the experimental observations. And we do find that salt concentration can modulate the direction of unwrapping as well as the asymmetry. And we were also able to look at some mutations. And with that, let me acknowledge Dave, who was my postdoctoral advisor. And much of the work was started at the fag end of my postdoc career. And also a very talented postdoc in Dave's group, Balaka. So she basically turned a very bad CPU code into a wonderful GPU code. And you can find, if you're interested, you can look up this. So our code is available on OpenMM. And thanks again. And I'll be very happy to answer questions. Thank you.
One or two short questions. Yeah, maybe one there. Yeah, please go ahead.
I wonder if you can focus on some of the lysines in the histone globular domain, if you can add acylation or...
Yeah, that is a very good point. So we are trying to now develop these force field parameters for this, methylations and acetylations. So that is it's still a work in progress. We're still working on it.
So you think your modeling can...
We have to test it out. I can't say a priori if it will work. So the idea is to develop those parameter sets and then see if it is able, it sort of recapitulates some of the observations. Yeah.
Thank you.
So in your salt gradient, when you show lower to the higher salt,
Yeah.
so there is some middle range, there are some specimens observed. What are those?
So those are basically partially unwrapped states where you can have the DNA coming from either one end
Okay.
or both ends actually.
Okay. But so there is no specific.
No, we find actually populations of both, but let me point out, if we did not quantify the populations as such, but we always find that as you go to the mid-range, it's always the asymmetrically unwrapped states which is higher in population, but we haven't really quantified that. Yeah.
Yeah, one question, yeah, quick.
So this asymmetry that you are seeing, left-right asymmetry, is it inherent in the sequence of the DNA? If you take palindromic, then you will not see it.
That is a very good question. We actually took a palindromic sequence. So this alpha satellite DNA is palindromic. So you have the same on both ends. So what we believe is, of course, the DNA sequence plays a role. If you have a stiff on one side, and so the stiff side will come up first. But here we also believe the histone tails are also playing some role, because what happens is because when you have a sequence same on both sides, stochastically one of them can come out. And then once one of the ends come out, there is some signal from one end of the one entry exit to the other. So there is some rearrangement in the histone octamer that we observe. So that basically pulls the other DNA close together. So in fact, this is consistent with some of the cryo-EM images at high salt concentrations that Halleck and others have resolved. But they also see similar kind of rearrangement in the histone octamer.
Okay, so let's thank Debayan and all the speakers who in this morning session. Thank you.